and welcome back once again to the fourth day of one week International Faculty Development Program on Herbal Medicine Research and Global Health, a therapeutic perspective, organized between 9 to 14 Feb by Adar Shikshan Prasarak Mandal, KT Patil College of Pharmacy, Usmanabad. Ladies and gentlemen, education is not the learning of facts, it's rather the training of the mind to think. With this beautiful quote by Albert Einstein, let's begin our new day of International Faculty Development Program. I hope you all are enjoying our different knowledge boosting sessions and getting benefits from them since 3 days and now ready to attend the session for today. Dear participants, if plants can provide food for our survival, they surely can provide medicines to cure our diseases as well. So let's go herbal and start a new day with new perspective. Ladies and gentlemen, recent advances in science have clarified the role of plant specialized metabolites, classically known as plant secondary metabolites, which cannot be considered only bioactive molecules used for human health but also pivotal factors for the global ecosystem. They play major roles in the plant life evaluation and mutualism. To provide a general view of plant specialized metabolites, it is important to consider both the biochemistry and the functional or ecological role of these important compounds. The field of plant specialized metabolism has witnessed tremendous growth in the past decade. This growth has had a profound impact on multiple disciplines in life sciences, including biochemistry, metabolism, enzymology, natural product chemistry, medicinal chemistry, chemical ecology, and evolution. It also has yielded valuable knowledge and technology readily applicable in various industries such as agriculture, horticulture, energy, renewable chemicals, and pharmaceuticals. The pharmaceutical sector is undoubtedly largely inspired by plant-derived compounds. In the second half of the 20th century, the interest in the plant-specialized metabolites in Western medicine, especially for those molecules which actually remain elective drugs for the treatment of several illnesses. So, for to know more about more aspects regarding plant-specialized metabolites in healthcare, we have one eminent personality in our expert panel today. Our resource person for today's session is Dr. Ashok Prabhakar Giri, sir, Senior Principal Scientist, Biochemical Sciences Division, Council of Scientific and Industrial Research, National Chemical Laboratory, Pune, who will orient us about plant specialized metabolites in healthcare. I would like to give a heartiest welcome to Dr. Ashok Giri, sir. Welcome, sir, and thank you very much. You have accepted our invitation and spared your precious time with us. So, before we move towards the session, I would like to introduce Dr. Giri, sir, in brief. Dr. Ashok Giri sir is currently working in the capacity of Senior Principal Scientist of Biochemical Sciences Division at CSIR National Chemical Laboratory, Government of India, Pune. He obtained his PhD degree in biochemistry in 1995 from Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. His current interests are molecular plant-insect interaction and plant secondary metabolic pathways. He is using omics approaches and validate functional aspects of specific molecules and genes involved in their synthesis. He has been awarded Alexander von Humboldt Fellowship and Max Planck
Miss Chitra,
Postdoctoral
we are not
Fellowship.
able to hear you. I don't know what is the problem.
Okay, sir. Wait. Hello, am I audible, sir? Hello.
We are not able to hear you. I don't know. It is a problem with me or you. I do not know, but we are not able to hear you. I mean, at least
Okay,
I'm
sir.
not able to hear you.
Okay, sir. Wait a minute. Hello, now am I audible, sir?
Yeah, it's better now.
Okay, sir. Sorry for the
I
comment.
think you have some internet connection problem probably. I don't know. There's probably
Uh,
internet connection. It shows that you might have. I mean,
I
ours
think
is
so.
looks quite okay.
Yes, I have changed the mic, sir. Now, is it audible, sir? Now it is clear
Yes,
audio, clearly
please go
audible,
ahead.
sir? Yeah. Thank you, sir.
Yes.
So, Dr. Ashok Giri, sir, has been awarded Alexander von Humboldt Fellowship and Max Planck Postdoctoral Fellow to carry out the research on understanding plant defense mechanism in native plant systems at Max Planck Institute Hello, can you hear me, sir?
Yeah, it is a problem probably with
Yes, sir.
internet connection,
We
not
are
with your
very,
mic.
yes, sir. We are very sorry for the internet connection. Now, am I audible, sir?
Yes.
Is it okay, sir?
Yeah, yeah, please go ahead.
Thank you, sir. Sir has been recipient of many fellowships, scholarship, and awards, including Fellow of National of Academy of Science, Fellow of Maharashtra Academy of Sciences in 2013, Scientist of the Year NCL Research Foundation 2007, Postdoctoral Fellow of International Agricultural Cooperation, Netherlands in 2001, McKnight Career Development Program USA in 1999 2003, Marathwada Gaurav Puraskar in 2005, CSIR Research Achievement Award in 1997 India. His major research credits include 175 publications in peer-reviewed journals like Science, Nature Communication, Nature Plants, The Plant Cell, Plant Physiology, Plant Biotechnology, Plant Molecular Biology, Planta, BMC, Plant Biology, Phytochemistry, Biochemical, at Biophysical, ACTA, Journal of Insect Physiology, Insect Biochemistry and Molecular Biology, Molecular and Cellular Proteomics, Journal of Proteome Research, Food Chemistry, he has also contributed 15 book chapters in the peer-reviewed books. He has 9 international patents granted and filed on his credits. He is PI of 21 research projects funded by national and international organizations. He has crossed 7,900 citations with H index 45 and ITN index 121. He has also been the convener of or organizer of various national and international conferences, symposium, meetings, and has delivered over 275 invited scientific talks in national and international conferences, workshops, DST supported Inspire camps, etc. He has supervised work of 10 postdoctoral fellows, 21 PhD scholars, more than 50 project assistants, and 75 MSc and MTech students during last 20 years at CSIR NCL. His current group comprises 10 PhD students, 2 postdoctoral fellows, and 6 project assistants. There is a lot more to tell about Dr. Ashok Giri sir, but due to time constraint, I will stop here. And sir, we have an indeed privilege to have you on our expert panel today, sir. Welcome you, sir. And now I would like to request Dr. Giri sir to start his session. Please, sir. Over to you, sir.
Thank you, Ms. Chitra, for nice introduction. I hope you are able to see my slides or no.
Yes, sir. Yes.
My slides are visible at the moment.
Yes, sir. Wait a minute, sir. Have you shared the screen, sir? Yeah, yeah. Now it is visible, sir.
Okay. So at the outset, I would like to thank Ms. Chitra and uh, the principal of the college as well as Dr. Shobha Tole for giving me this opportunity to interact with the uh, all of you, especially students and faculties who are very enthusiastic. And I'm sure in last 1 week you must have been, I will say that got a good feast from several good speakers, which is the again program organized by your college, which is called I think the International Faculty Development Program, and which is also on a very important area, herbal medicine research and global health, a therapeutic perspective. It's my pleasure to be there with you and share some my views and whatever the work, small part of work I can present here that going on in our group. So I would like to start my talk with a beautiful say of Sant Dnyaneshwara. I think Sant Dnyaneshwara doesn't need introduction to all audience. So he says, I mean, he was there in 13th century, and he says, "Zhade olkavi, phule jana olkavi bole." And this is, I mean, I feel it is so great because, you know, after a lot of research and so we came to know that if you want to identify correct plant, since the workshop is on the plant, when my talk is on the plant metabolites, so to identify a correct plant family and its classification, the flower is most important character. And this we came to know maybe probably last 200 or 300 years research, and we now know that flower is the most important character. But if you can imagine the visionary person like Sant Dnyaneshwar, he said, if you want to identify a plant, you need to understand its flower, and that may be able to help you. Whatever the other part, Jana Volkhave Bole, what he said is also true probably that time. today probably it is not true because people talk differently and act differently. So it may be not true for human beings, but it is definitely true for the plants. Not only that, I have another say, and this is again related to today's talk. He says, Nagarechi Rachavi, Jalashaya Nirmavi, Mahavane Lavavi Nanavithe. And this is also inspires me a lot because he is not against the development. He is actually for the development. But one has to understand between the lines, he did not say that you should plant eucalyptus tree or any specific tree. He says Mahavane Lavavi. That means he understood very well what is the biodiversity and what are the actually plants that we grow in the various regions. And he has not said that you plant one specific tree or a few specific tree. He says Mahavane Lavavi. And that for that, we have to understand that he also knows the diversity and importance of the diversity. And this is again says Nanavide. So this is how our culture and our, I will say, philosophical people we were privileged to have in our country, in our region, or in our, I mean, on this part of the world. So why I'm saying this, because I will always gets inspired with this kind of a philosophy, and I mean these are, I mean, I will say very well great science, in fact, which is behind it. So it's, I will not say that this is not science, because if he says that we have to identify the plants based on flowers, so it was very much science as well, what today's science we talk about. So by getting inspired with this talk, we had a very interesting project at NCL, a very small project, and I'm introducing you with a very specific reason, and which is called native forest, for example, which is actually a compact plantation. And this is an ecological experiment to replicate the forest, Western Ghat, part of this Western forest in the small area, which is not more than, I will say, 10 gunta. And our I will say honorable minister that time, Dr. Harshvardhanji, came, and we have planted one of the plant with him, but we have more than 100 plants which we are growing there. And this is one of the jungli hirda. Okay, and you know, he is our ex Minister of Science and Technology and few other responsibilities, and is our the NCL director that time, Professor Nangia. He is our DG, Dr. Shekhar Mande, and several of my colleagues. So we planted this tree, and why I'm showing is, for example, when we looked into this area and developed little bit, so it was like that. And I have taken this picture last year in the lockdown, and you see how the diversity that actually popped in this area. And it's not only the plant, but by the example, if you see, there are several important plants which are growing in this 10 guntas. But then what is more interesting, and this is one of, I mean, I take some PhD coursework, so the students actually try to understand what are the different insects that visit on these plants. And we could see that these insects are coming and actually enjoying their food, even shelter, and everything. So one can imagine, like, this, some of these insects, because these plants are not actually typical what we grow in our garden, these are native plants. And by saying that, there are also native insects which come there. They are specialized insects, and we could see, they could see that there is a one plant growing there, and they went there. So these are like something we called it as a monophagous insect. That means they grow, or they come to visit a plant, or a specific plant. And now you will, I mean, this is a big question for us, that how these insects identify, or they know that this plant is there. And for that, actually, these metabolites or plant metabolites are basis for these interactions. So plant and insect or any organism in this nature, they talk a universal language, which is called chemical language. And whatever the chemical language that they talk, it is actually very responsive, and that's why these insects are coming and visiting this plant. So that's the beauty of, I will say, nature, and that the beauty of the language that nature talks. Because you know what language we talk, probably if you pass maybe few hundred kilometer area, it may not able to, I mean, I will say, understand by somebody else, or neither we understand something from them. So this is the nature's language, and one has to understand this chemical language, or the plant metabolite language, or insect metabolite language in a better sense. And that's what we are trying to do. Since I mean, all of you are, I will say, young researchers who are performing your respective degrees. But when you come to the research, I feel that research is beyond borders, and this is one example I'm trying to give you. And this is Dr. Venkat Raman, who got Nobel Prize in 2010. And I was just trying to look his background, and it was very surprising for me that he did his BSc, MSc, as well as PhD in the physics. Okay, and then when he entered into the research, he started working into the, like I will say, how the uh, uh, protein biosynthesis happens in bacteria, and how these ribosomes recruits specific tRNAs and so on. So that was his research. So he did a research in biology, studied in physics, and probably Nobel chemistry did not realize that he doesn't have a background of chemistry. He got a Nobel Prize in chemistry. So I really want all of you to give the message that, and this is not the only one story, that when you come to research, there is nothing like this subject boundaries you should really cross, and you should work beyond borders. And this is not the one example that I'm trying to give, but since, I mean, everybody knows Nobel Prizes are the one of the best prizes in the world for any science that we do, and they are for several disciplines. And just to continue it further, you see, I mean, I just reviewed it last like 12, 13 years Nobel Prizes in chemistry, and you will see all these red. For example, discovery of the green fluorescent protein, or just now I shared about this structure and function of ribosomes, or studies of G-coupled receptors, or mechanistic studies on DNA repair, or maybe last year, like 2020, the Nobel Prize goes to this two young women scientists for the work on genetic scissor, which is actually called a tool for rewriting the genetic code. And you can imagine, and it will be surprising for some of you, that chemistry Nobel Prize goes to the molecular study. So that's how the research is beyond borders. And this is what I thought I could share with you. So now coming back to this plant metabolites, and as Chitra nicely introduced, that these plants actually produce these metabolites. And as a human beings, I mean, we are always, I will say, very much biased, and we always give this terminology. We say medicinal plants. In my opinion, all plants have some or other medicinal properties, but still, based on our understanding, we have categorized that there are some plants which are medicinal plant, and we'll talk about that only today. In some of the plants. So again, when, whatever you say, for example, so a particular compound has anti-diabetic properties, or a particular compound has anti-cancer properties, one has to imagine that why plant will produce something which is anti-diabetic or anti-cancer, or whatever the diseases that we human being gain. So these plants has lot of, I will see, these metabolites, and as all of you know, plant are, they cannot move, so they have to sustain in under a given conditions. It's a do or die, and for that reason they have a lot of, I will say, chemical diversity or metabolite diversity that helps them to really manage in a given atmosphere. It's not only diseases, or it's not only insects, or it's not only drought or salt or so. Whatever the stress they get, they have to manage with that, and that is the reason why they have called, I mean, they could survive, and that's how there's a lot of diversity, and that's how they produce these compounds. And again, they don't produce these compounds all the time or in all the tissues. There is a specific mechanisms, and these mechanisms are very much sophisticated mechanisms. For us, it is a bit complex to understand, but they are very well organized to produce these secondary metabolites. And in our, I mean, if you think about even just India, we have this Ayurveda, Yoga, Unani, Siddha, or Homeopathy, which is, we call it as the Indian traditional medical system, or Ayush, has been actually described the importance of several plants, which I mean, you could see the images up here. And today, I mean, I will say more than 35 to 1,000 to 70,000 plant species are used worldwide for various reasons. And some of them do have established their medicinal properties. And what I said is, actually, these are actually plant producers for a number of things. And even if you go beyond that as well, if you see the descriptions, for example, whether it is China or India, it has been discussed, maybe probably even 2500 BC, or more than 5,000 years old, like how people are using various plants, and plant products or various things for treating the human diseases. And it is very much there in the Vedas, and then there are more than 700 plants described in the Chark Samhita or Sushastra Samhita. That's what we know. So if you think about today's drugs, for example, and I will give a couple of examples, and I will say these are like really proved to be the very important for the even today's, we called it as allopathy or modern medicines. So these are the molecules. Several of these molecules are used for these properties. Uh, Being the produced by the nature, of course, it has a lot of, I will say, advantage, and that's why this economy runs with this. And there are a lot to discover. I will say there are plenty of them are available, and one has to really understand in various angles. And just to list out few of them, for example, everybody knows this Respirin, which is isolated from Sarpagandha called Rautia serpentina. Or there are several alkaloids, like for example, this morphines or nicotine, or even several opiates. These are actually coming from various plants, and they have been proven to be very important. And maybe all of you probably know this artemisinin, which I'm going to describe probably in the next slide as well, that it is produced by this plant called Artemisia annua, which is a sesquiterpene lactone. And this is only one drug. I will say the artemisinin and its derivatives are the only one drugs available today to treat the malaria caused by this Plasmodium falciparum. So, if you think about it again, what are the importance of this medical plant? So yes, there are, I will say, a lot of significance to it. And when I was looking into the literature, whether it is on Wikipedia or Google, I could see that such information is available, and we will talk a little bit about each of them. For example, there are more than 110 molecules which are plant based and which are currently used as a drugs. Some of them are derivatized and so on and so forth, but these are actually used. And take example of as simple as metformin, for example. So if you see the history and if you see the today's market, it's amazing. And again, it is coming from the plant. Not only that, all of you know the Taxol, for example, coming from this plant, which is again chemical used to for the cancer treatments. And we have understood in several cases the mechanism. I will say not completely we know how these work as anti-cancer compound. Similarly, like for example, just now I was mentioning this artemisinin, which is actually got a Nobel Prize to this lady, again a Chinese lady, Professor Yu Tu. So she started actually understanding probably this plant, which is used in the traditional Chinese medicines, as well as even it is used in even our traditional usage to treat the fever. And this is a magic drug coming from this traditional China medicine. And again, as I mentioned, it actually works against this Plasmodium falciparum. Of course, the detailed mechanism is not known, but it has been used today, artemisinin or its derivatives, very successfully. If you think about aspirin, it's the same story coming from this plant, Salix alba. If you think about this other drug coming from this another plant called Digitalis lantana, which is also, I will say more than, I will say close to 100 years. And if you think about even future drugs, for example, we talk a lot about curcumin coming from our haldi plant, and maybe in the combination of several things. So what I am trying to tell you that there is a lot of scope to really discover new molecules and to bring to the stages like this. Only one message to all of you I wanted to say, that it is a long process, and one has to have that patience to work on a specific thing and dissect out so that we can get a recognition. It's very unfortunate that none of these discoveries are coming from India, although we have this traditional knowledge with us. I will probably try to tell one story that we are looking into that direction, like how we can really come up with a drug molecules, not only the herbal but drug molecules, which is coming from the plant and could be successful for something like this. And for that, actually, for this is a slide actually today morning I made, because this is a paper today published in Chemical Sciences. It's not a paper; it's a review. And the title of this review is "Natural Product for the drug discovery in the era of artificial intelligence. And what I wanted to, and why I have put this slide early morning today, is that you guys, I mean, several of you are like students of BSc or MSc or B Pharm or M Pharm. So you see, this is a girl who just completed her BSc, and this is a boy who completed his MSc very recently. And they could come up with this review, which is published in one of the best journal in chemical sciences, having impact factor 9.2. Why this doesn't happen in our country? And when Ms. Chitra was reading my CV, I mean, you must have been, realized that I visited several countries and I had opportunities to work not only with the senior professors like this, but also I have a lot of opportunity to work with students at BSc level, there are undergraduate level or postgraduate level. And when I see the understanding, the way they think, the way the faculty gives them exposure is completely, I will say, amazing. And I feel sometimes very sad that we don't give such exposure to our students. And what is this actually? In fact, a couple of years back, when I gave a talk to faculties in Pune University, I said that this machine learning and artificial intelligence is something very important for new generation people. Maybe for us, it may be a little bit difficult to learn these languages and so on and so forth. But I think a student of this era should have that, something like you learn basic languages like Marathi, Hindi, English. So we should also try to understand this language, which is going to help you in long term to give a different perspective to your research career. And what this is actually, what they have reviewed, by the way. So they have categorized them into three things, like computer assist discovery of natural products and how one can do the extraction, what I will try to tell you, and how they analyze these metabolites, and how, I mean, all the data is sometimes available in the various database, various scientific databases. And at the same time, we also know a lot of information is coming from this next generation sequencing, whether it is genome sequencing or whether it is a transcriptomic sequencing or even beyond genomics. I mean, the tools that we are developed, like for example, genome editing or genetic manipulations and so on. So if you have this combination, you can come up with several compounds. You can do a lot of screening based on bioinformatics data, and taking care of all proteomics, metabolomics, genomics, interactomics, and come up with activity. And then you can have both spaces, like for example chemical space, where we can even modify these molecules, come up with a different derivatives, and as well as at the same time you can learn, or you can develop a tools using synthetic biology or protein engineering or enzyme engineering, and come up with the compound, come up with the enzymes or the scheme which can produce even not only the natural products but then derivatives, and finally this can have a lot of biological significance. So for all these, at various stages, you need to have a good background of biology, good background of chemistry, and good background of bioinformatics analysis, and then you will be able to build such a pipeline in that case. So what I mean, why I did put this slide is basically to show you that yes, it doesn't matter whether you are undergraduate or postgraduate student, you should come up with such ideas, and you should try to explore either entering into the research or writing reviews, so that you can have a good understanding. And we did this, I mean, I'm practicing this from last 15-20 years. For example, this is one of the review last year we published with one of my PhD student, Tanuja, and he is my colleague Dr. Bhushan Dolakia, and myself. So what was this review talks about? Because all of you know our holy plant like osmium, which we have this osmium telliflorum. Earlier we used to call it as osmium santum, but then we know that there are more than 50 different species of osmium, and there are maybe more than few hundred subtypes or chemotypes of these osmium species, and which has a great significance. And what we have reviewed in this review is basically what are the trends, implications, and strategies to improve the quality as well as the yield of essential oils. And these essential oils has great significance in our life, or even whatever the modern lifestyle that we have. And we have reviewed that which are these metabolites there, which are this, what are the role of these volatile compounds in the plant, and how we are actually exploring it further. What are the reasons for this chemodiversity that happens in the nature, and how you can even further use to do something what I just said in a minute before, that produce such compounds in a very high amount, and then how can overall we can increase this either in the plants or even in the in vitro culture, or maybe we can even do this in the fermenters using various kind of yeast or bacteria and so on. So this is what we have reviewed, and again, till this point, I mean, we with my group we have written something like more than 25 reviews, and there are few reviews in the pipeline. That gives you a very good understanding that what's happening in the in a specific field, and then students come up with a very nice questions or genuine questions which has a high impact in a longer run, and then that's how they continue their research activities further. Okay, so this is just another example that happened in the first lockdown. For example, most of our labs were closed in March, and we do not work on actually developing molecules for, I will say, a specific antiviral or so on. But then there was a pressing question, and then there were a lot of information available about this COVID-19 targets, which probably people were thinking that time could be a potential targets to develop a drug. And in the when the first lockdown happened in 2020, I mean, we have the library of more than, I will say, 7,000 or even 10,000 compounds, which are coming from various sources, for example, plant-based, or even we have used some of the antivirals as well. And we screen based on this is all when labs were closed. This all happened using bioinformatics, and that's a good team headed by my colleague Dr. Rakesh Joshi, and we come up with several molecules which are potent, I will say, inhibitors of one of the important protease that present in the COVID. And other than RNA-dependent RNA polymerase or this angiostatin converting enzyme as a target, even the MPRO, which is a main protease, is also used one of the target, and we could come up with some molecules from plant-based or antiviral-based, or the molecules which are in the, I will say, lead molecules. We could come up with this analysis, and a journal which is called Journal of Biomolecular and Structural Dynamics, they accepted our paper within 5 days, and they published on top priority. And in last, I will say, 1.5 year or 2 years, this has been cited more than 225 times, and some of the molecules people have taken further. And we also actually explored this possibility, and we also got DCGA approval to do the clinical trials of these molecules. Unfortunately, it was a bit delayed, but still we got very interesting data. So this is what I'm trying to show you, that okay, you need to have open, and you need to increase your skills to be, I will say, competent in a current time or recent time, or even in future. So let us talk about like plant metabolism, okay, or plant metabolites, which we called it as a specialized metabolite, or I will say secondary metabolites. So if you think about its size, for example, in yeast, we have plus or minus, I will say, 550 compounds. But if you think about a single plant, I mean, the result shows that we have maybe more than 20,000 compounds, and more than 2 lakh structures are identified from the plants, and there are actually probably a million metabolites coming from 270,000 different plant species which we have. And these metabolites, again, broadly, it is I mean, they are actually coming from these 4 classes. And what are these 4 classes? Basically, all of you know, one of the metabolites based on their structure and so on, it is called terpenoids or isoprenoids. And again, there are subcategories like monoterpenes or sesquiterpenes or diterpenes and tetraterpenes, and then it also gets further complicated. So this is also a very important class of terpenoids. Some of these molecules are also volatile, and that has a lot of even significance in the aroma, as well as in the fragrance, as well as even other important characteristics. And again, we are exploring these molecules even for human usage or even the other uses. The another class of molecules is called alkaloids. Again, I think I have given you some examples of, like, for example, caffeine also, and they are produced from various sources that I will talk to you little bit later. But then this is also a very important class. The third class, which is called phenylprepenoids, again, it's a complicated molecules, and several of these molecules have great significance as far as the plant's physiology is concerned, but then we are also exploring these molecules for our usage. And the fourth class is the peptides, which is actually new. And one has to think that these molecules, the peptides, might be also has a lot of significance not only for the plant, but even if you think about its application for any application that as a human we can think about. So the core question that scientific community is asking today is how and why this diversity is formed. I think I have answered part of your question, part of this question. For example, like plant has to survive in the given conditions, so probably that is the reason. And but what is more interesting for me as a scientist, or for all of us as a researcher, is to understand this nature's chemistry protocols. How this large diversity is synthesized so precisely, and not only it is like a now stop, but then there are also we are discovering new metabolites, or maybe even there are new metabolites are formed, and thereby synthetic pathways are getting involved. So these are the core questions that scientific community is asking, and then we are trying to understand it. So what are the reasons? Again, we have reviewed little bit about it, that why these metabolites are produced. So of course genes are responsible for that, but then how these new genes are actually new functions are coming, and then that's why we see this large diversity. And then the reasons could be mutations, could be gene duplications, or maybe allelic variations, or even for that matter there is something called de novo functionalization is also happening to generate and to maintain this diversity in the plants. So how this biosynthetic processes happens, and these are very very precise, and they are well organized, not only in the specific tissue but within the plant cell. There are several organelles, and all of you know that, for example, if you think about plant cell, we have chloroplast, we have mitochondria, we have vacuoles, we have cytosol, we have and so on. So all these organelles actually are very also well organized to produce something which is required for the overall plant. And this is well organized process. For example, this melanite pathway happens in the cytosol, which gives you IPP, which is a building block for terpenoid synthesis, or to get this terpenoid diversity. On the same side, that there is something called chloroplast or plastid, which is present particularly in the plant cell and some bacterial cells, but that also gives you IPP, but that is happen in the plastid, and this is another pathway which is called MEP pathway. So you can understand how this precise processes happens in the plant, even to generate the similar kind of molecules, and which are actually building blocks of this, I will say, terpenoid biosynthesis. And all of you know this. Like, for example, if you want to have monoterpene, which is a C10 molecule, it has a substrate called IPP, and that IPP get isomerized with another molecule, and then you have some enzyme, some basic enzyme in the monoterpene biosynthetic pathway is called GPP synthase, which use GPP as a substrate which works for, I will say, monoterpene biosynthesis. And these monoterpene syntheses are actually well known. There are several of them in a particular plant as well. Not only that, this GPP also works the substrate for FPP, and again you can imagine now we have 3 IPP units. That means for this sesquiterpenes, like, which is again FPP synthase gives you FPP, which is again substrate for sesquiterpene biosynthesis. And then and these sesquiterpene synthetases are actually well known, and they have been giving this lot of diversity in the sesquiterpene, and this is followed by diterpene and so on and so forth. So what I'm trying to say, that how this biological processes or enzymological processes are well planned, and not only in the specific plant or specific tissue, but even within the cell, they are well organized. This is how, it happens for the terpenoids. But if you think about alkaloids, which are much more complex molecules, so the speciality of alkaloids is that it has to have nitrogen. They are basically derived from amino acids or nucleotides, but that's not the all. There are some molecules which have actually alkaloids which are coming from even different compounds. And then one has to understand their structural diversity, because there is something called tryptophan derived monoterpenoids, like, which is, we called it as a indol alkaloids, or arginine based derived alkaloids we call tropane alkaloids. Or we know caffeine, for example, it comes from nucleotide derived alkaloids. And cholesterol derived alkali is called steroid glycoalkaloids, which are also have a lot of significance. Their biosynthetic pathways are much more complex. For example, you need a lot of broad specificity enzymes as well as very specific enzymes, and their functional characterization is challenging. And what is very interesting, which came out in last 10 to 15 years, is whenever we see these genes which are encoding for these enzymes, which are broad specific or even very specific, they are actually sometimes clustered on the chromosome. That means they are present within the small region of the chromosome, and probably that helps to regulate this biosynthetic pathway in a very precise way. And there are various protocols actually be used in the lab to characterize the function of such molecules. So to go it further, for example, this alkaloid biosynthesis pathway could be much more complex, because one cannot really, for example, and this is the work that we are working together with Weizmann Institute of Science, for example, to understand the uh, steroidal glycoalkaloid biosynthesis in tomato and potato and in eggplants, and it took a decade for us to understand, I will say to some extent, not even completely, the how this arpatomatin or arpaspharanin is produced in the tomato and potato respectively. And then when we actually try to answer this question, we used very systematic approach to understand, first of all, that what are the substrates. For example, cholesterol is a substrate, so how cholesterol is produced in the plant. Then from the cholesterol, how this tomatidine, which is a glycan form of steroidal alkaloid, is actually formed. So once this tomatidine is formed, how this is decorated with different kind of sugar, whether it's galactose or glucose or xylose and so on, how this happens. And then how this molecule is further even modified, something called esculoside A, which is actually, for example, alpha tomatin has some, I will say, bitter taste and so on. How this compound is actually getting metabolized to form a safer compound for human health, and how this happens in the natural processes, whether it is a wild tomato or cultivated tomato, or it's a wild potato or cultivated potato. So we have to use various tools to understand this SCA pathway, and probably I will say that we are more than 20-25 people working probably from last 10 plus years to unravel this complexity of steroid glycoalkaloid biosynthesis in the these two plants species. If you think about another molecule class called phenylpeponides, which are also very important, and I'm giving a very small example here, like for example, how they are synthesized, and they are also much more complex. For example, several of them starts from this L-phenylamine, and this PAL enzyme has actually multiple isoforms, and they also have a specific function. And then this pathway starts, and which is actually, I will say, it's much more complex, because it can generate something called lignin, and some of the part of these compounds go to even biosynthesis of a specific compounds called estragol or eugenol methyl ether and so on. And then some of these enzymes are quite common, broad specificity. Some of them are very specific for to produce a specific metabolites. And to understand this, it looks quite easy to now explain here, but it took substantial time for us to understand how this happens in a particular plant species, and it's still actually, I will say, it's going on. So one thing we realize, that although we called it as a specialized metabolite or secondary metabolites, but the basis of them is actually always starts from primary metabolites, whether it is a glucose or whether it is acetyl CoA or whether it is some hormones or some nucleotides and so on. So the bridge between primary metabolites and secondary metabolites is not actually, I will say, there's very nice crosstalk between these molecules, and that's how they decide whether what to synthesize and how to synthesize and how to really create such a huge diversity. Sorry. So now question is like, okay, how to analyze these metabolites? For example, here we know now there is a lot of diversity, and we need to really understand this diversity in the form of isoprenoids or alkaloids or terpenoids. But how to analyze this? And that comes back to the question like, okay, how we analyze these molecules? So it's a very, I mean, there is no any specific method available to extract all the metabolites in any of the solvent system or any of the, I will say, extraction media, if you would like to say that. There's no really a universal solvent system available or extraction media available to extract the metabolites. So it's always, if there are 2000, maybe you may be able to extract only few of them, and then question is like how you analyze them. So very classical method is, of course, I'm sure all of you know, thin layer chromatography or HPLC or paper chromatography as a primary thing that we use. But more advanced things are happening probably in the mass spectrometry based methods, whether it's GCMS or I will say LCMS. And these are the very, I will say, high-end methods or tools available to unravel the diversity of these metabolites. And something which comes out from the particular plant tissue or any tissue for that matter, that means these compounds are volatile. So there are very nice methods available today to extract or to analyze the compounds which are volatile, which are coming out of the particular tissue in the air. Of course, there are limitations, whether it is TLC or GCMS or HPLC or LCMS. So, for example, for HPLC, unless you have standard, you cannot really identify, or you cannot. It's not easy to know or find out unknown compound if you analyze on LCMS. If you want to even go further, for example, GCMS, so the good part of GCMS is there are several libraries, that means molecular libraries, more than 3 lakh compounds are available in these libraries. So if you analyze any sample, you can see, okay, within this library, whether my compound is there or not, and there are nice tools available for that. But if the compound is not there in the database, you may not able to identify it. And for that, this method, which is called LC-MS-MS based, or this is called tandem mass spectrometry, basically. So you can even fragment the molecule further, and you can identify it. But for that, you need really a good background of a chemistry, whether you want to use spectroscopy or whether you want to use mass spectrometry. For both, you need a good background of chemistry, and that's why you should not forget chemistry, although you are in the pharmacy or even I will say other sciences. Like, for example, I am biochemist, but I have to remember this chemistry, then only I will be able to identify. And these are the areas, actually metabolomics and metabolic pathway analysis is actually a promising areas in the area of chemical biology, or if you would like to say synthetic biology, which is also true. For in both cases, this mass spectrometry is going to play a crucial role. And there are several reviews, if you go back and see, that they talk about these methodologies, and I mean, I will say novel things coming from this methodology. I'm showing this slide with a specific reason, because the students coming from area like ours, I mean, I also coming from a similar district like your neighboring district called Bede, we get always scared of these high-end technologies. But I would like to tell you with this slide that all these high-end technologies are based on very simple principles. So, for example, this lady, she is trying to separate, and I think all of you know what she is doing. So she is trying to separate the grains from other thing. And what, if you think about it a little bit scientific language, she is trying to separate this based on their molecular weight or weight. And exactly this same happens in the mass spectrometry. So if you understand this principle correctly, and whatever the sophisticated instrument that you use today, they are very simple, and don't get scared of them. If you know how to make bhakri or chapati, or how to make khichdi, or how to do, I will say, some work in farm, I think most of the techniques are much more simpler than that. Only thing is we get scared, and I think we sometimes don't get access to use these kind of techniques, and that's why we get scared and we feel that it is too complex. The most I will say most high-end equipments have minimum to do by ourselves. A lot of things are available in the system itself. We have to analyze, and what is most important, in my opinion, is to really make sense of that data what you get from the particular analysis. And for that, you need to have your good reading, good background, and good analytical power. So this is another way, as I said, one can do the headspace measurement. Analysis, for example, you can keep something in the closed vials, airtight vials, and whatever is released from, particularly, for example, from this leaf, you can analyze with this SPME, which is called solid phase microextraction, or one can think about trapping, because you pass a clean air, you have inlet and outlet, for example, and you put something here which this volatile molecules get trapped, and once your experiment is over, you can extract the metabolites from this trapped thing which came in the air, and then you can analyze with a suitable method, whether it is from particular tissue or it is from particular plant or whatever the sample that you have. So my exposure goes back probably long. When I went to my second postdoc in Netherlands, I started working on this very interesting plant system called strawberry, and what we could really gain is like when I took a cultivated strawberry, what we eat, and wild strawberry, and we could see simple, like this is a simple GCMS analysis. I could see there are two molecules, for example, linalool and neraldal, which are very prominent in the cultivated strawberry. This is a monoterpene and this is a sesquiterpene, which is popping up quite nicely here. Are there only in the cultivated species? That means what strawberry that we eat, for example. And this is a wild strawberry, which is called Vesca, Fragaria vesca. And when I see this careful analysis, the compound that we see, for example, linalool and neraldal, are not here. But what was more interesting is this strawberry also have monoterpenes and sesquiterpenes, but they are different. And based on that, we come up with a idea. That means, for example, the how the strawberry is actually came into the domestication. That means maybe probably 2000 plus years, that we have developed the current strawberry what we eat from this strawberry. And during this process of domestication, that means the way we started cultivating and so on, we have also did the, I will say, genetic engineering or metabolic engineering to have these compounds in the strawberry what we eat, and to get rid of these compounds which probably are not so good as far as the aroma or taste is concerned. So if you can imagine, this metabolic pathway engineering is not today's time, although we know now how it has to be done more precisely, but we are doing since our civilization. So whatever the plant species or crop species that we grow, this went under such a, I will say, genetic modifications what we see, and that's why today we enjoy, I will say, sweet and tasty and nutritious food. This all happens because of this genetic engineering. This is how one can do, like for example, this is another tool. This is a tool that what I showed you, like GCMS, and this is LCMS based analysis. And you can imagine, it's not only that you identify the molecule, but you can even do absolute quantification or relative quantification of these all metabolites which are present in the potato, either tuber flesh or tuber peel or whole tuber or roots or even the stem, like underground stem or upper stem, or whatever the tissue that you have. For example, these are different parts of the flower, like ovaries or buds or anthers or stigma or petals. So one can analyze, and this is what, when I was in Israel, I was analyzing these metabolites from the potato, and we could see there is a lot of, I will say, interesting molecules which are key there in a specific tissue. For example, if you see all this red, means these compounds are very high in the flower tissue compared to the other tissue, and other and there are like the compound that I am looking, some of them are also present in the tuber flesh or tuber peel. So I will try to give you some examples. I don't know how much time I have. Miss Chitra, you can tell me how much time I have? Hello? Hello? Are you there?
Yes, sir, yes.
So how much
Yes,
time
sir.
I have?
Up to 1 o'clock, sir.
No, but I want to have some question answers session as well.
Two
So,
hour session is there.
sorry.
Yes, sir. Huh.
So maybe yes.
Up to
Okay.
1 o'clock, we have the presentation, sir. Yes. And
So
after that,
that's
we
not
have
a
question
problem.
answer
Okay.
session. Yeah, yeah.
Okay, so I will try to give you some examples which already just now I talked about. The very first example is like the strawberry, where I got exposed to work on this specialized metabolites or the metabolites which has good nutrition value as well as taste. So I will give the first example, which is actually a strawberry, and then maybe when I came back to India as a scientist, I got a job as a scientist in NCL. So I started working on initially on mango. That is very important plant species for us. all of you know, compared with strawberry, for example, mango is gives a lot of I would say foreign currency to us, as well as we also use these mangoes for our say nutrition as well as other things. Then we started working on with my colleagues. We started working on osimum. Again, I'm going to tell you a small story of it, that how we come up with a specific molecule from osimum. Potentially, it could be used for the management of the diabetes. Then, if time permits me, then I will tell you these three stories. For example, steroidal glycoalkaloids from food plants, which is potato and tomato, which are anti-nutritional compounds, and how we can get rid of them by using, I will say, modern techniques. Our even, I mean, you don't have to use you not necessarily modern technique, but even can think about using even, I will say, classical approach to get rid of these compounds once you have understanding that how these molecules are synthesized. Then this is an exotic plant called Ceropegia, and they produce a very specific molecule called serpigines, which are again alkaloids, and it is not yet known. But if you see traditionally, like our tribal communities, they use for various things these metabolite. I mean, these plant extracts actually, or even plant parts. And then question is like how we can use, or how can you produce these molecules in a large amount for various applications. So if time permits, I will cover last three, but otherwise I would like to have a question answer session, so that I mean, it will be much more interactive. So coming back to this same slide, what I showed you, like this is what I think, I don't want to spend again time, that you must have been understood what is meant by gain and loss. During the process of domestication, you could see that these compounds are gain in the cultivated strawberry. That means the strawberry we grow in the field, whereas these compounds are lost. And that time there was not like really NGS was available, but microarray was very routinely used. So we used this microRNA, and we could identify the specific genes or transcripts, I will say. In that case, they are expressed in the I will say the strawberry, which are actually started to get ripen. So all of you should know that strawberry ripens on the plant. It's not like a mango. So when you think about these specific genes, what we have isolated, or the specific gene, which called it as an neuraldal synthase, if you see, it is expressed only in the fruit, and not only that, but it is not there in the green fruit, but it is expressed in the ripening stages, and it is very high in the red fruit. So that means the compound that I showed you in this slide, for example, so are probably responsible, or they are actually synthesized by this gene called neuraldal synthase gene, because we could see that neuraldal is there and linalool is also there. So this gene we characterize further, but this also gave us an indication that yes, this gene is actually could be responsible for making this compound. And then we of course did a lot of its molecular characterization to express them into E. coli, and we could demonstrate yes, it do produce. For example, if you give GPP as a substrate, and this is the linalool, for example, reference R linalool and S linalool. And if you give the enzyme, which is actually the gene was taken from the strawberry and put into the E. coli and got the recombinant protein, and if you incubate this recombinant protein with a GPP or GDP as a substrate, you could get a very nicely S linalool. And if you see in the strawberry, what is there in the strawberry is of course S linalool. And this is where we can actually characterize the particular gene in a very simple way. There are much more complicated ways to also to do it, but if you are lucky, you are really synthesize. And this is nice correlation that. And the beautiful part of this is these enzymes are very specific. So one of the challenge in synthetic chemistry is that we don't have control of chiral forms. So for example, whether we can get R form or S form, it's very sometimes tricky. So you might get majority S form or I form with chemical synthesis, but there is still a possibility of getting the other chiral form. But if you enzymes, you have zero form. So for example, if you see with recombinant enzyme, you have not even a traces of R linalool present, and what we see exactly that in the strawberry. So that's the beauty of, I will say, biological processes or enzymes which are present in the plant. So what we did further was we looked into the question that we were asking: what is the why these compounds are gained during the process of domestication, and why these compounds are actually lost during the domestication, which are present in the wild. So for that, we analyze more than 15 cultivated species of strawberry all over the world, and 15 wild species which are all, I mean, collected from all over the world, and analyze its genomic composition as well as the expression. And what we could see very nicely, this particular gene which is responsible for making this linalool and neraldol, which is seen in the cultivated species, is very specific for, I mean, specifically expressed in the cultivated and not in the wild. And that gave us, I mean, I will say, nice clue why I will say these compounds are there in the cultivated strawberry. Further, when we look into it, we could see that if you see a typical open reading frame which gives you a protein, so it has either signal or target peptide, and then we have mature protein. This is happens in lot of cases actually. And this signal or target peptides could be actually specific for actually a specific compartments, whether it's a chloroplast or mitochondria or vacuoles. And if these signal peptides are there or target peptides are there, so that decides the fate of this molecule. So when we looked into the gene which we cloned and characterized from the cultivated strawberry, we could see that there is a truncation in the signal peptide or target peptide. That means the typically this monotropin synthase gene, which produces linalool or in this case neraldol as well as a sesquiterpene, it is supposed to be present in the chloroplast. But then what happened probably in the cultivated strawberry, when we looked into the detailed sequences, we could see in the cultivated strawberry either there is a stop codon in the region of target peptide, and to also there is another methionine actually present in this cultivated strawberry gene, which is coding for neural synthase or neural synthase. So what we did to prove this further, we fuse this gene construct with green fluorescent protein, which is actually classically used to study localization. So, and then we also predicted based on the programs what we have, and it was quite nicely matching. For example, if you take a target peptide from the wild strawberry, it shows that it has a very nice targeting for the chloroplast. So if you see, all this is actually chloroplast, and this your GFP is actually getting fluoresced right there. So it's not there anything anywhere else, whether it is a plain image or stack image. So this is typically signal that is present in the wild strawberry. If you take a sequence, whether I said that either there is another mutation between methionine 1 and methionine 2, or there is another transcript that we could see very nicely expressed starting from methionine 2, and if you fuse with GFP, we could see that now the chloroplast targeting is already gone. So this GFP is present in the cytosol. If you see very nicely, it's not in the chloroplast. These are like autofluorescence of the chloroplast, red we see, and GFP is always not in the chloroplast, but it is in between. So that itself gives us an idea that during the process of domestication, the gene is there in the wild strawberry as well as the cultivated strawberry. But in the wild strawberry, since it has actually a target peptide which takes this protein to the chloroplast, probably that gene is not expressed in the wild strawberry, and this gene is very well expressed. And I showed you the Western blot in earlier slide. For example, here if you could nicely see that this gene is very well expressed in the cultivated strawberry. That means if there is no target peptide, this gene is getting expressed. And that's why the literature search is also very important. For example, when we looked into the literature, we saw that this group has published in 1977 that what happens in the strawberry ripening stages of strawberry. And if you could see nicely, that plastid is there in the petals, for example. But then as soon as the fruit starts actually ripening, this chloroplasts get degraded, and especially if you see in the red fruit, these chloroplasts are actually completely disintegrated. And now you put both data together, the metabolite data and gene expression data, and then signal peptide analysis data or target peptide analysis data, we could see that yes, of course, the gene is not expressed in the wild strawberry because the gene has a target peptide which says that it should go to the plastid, but there are no plastids. And this is what we story published in Plant Cell in long time back, saying that it is a gain and loss, and why, what is the molecular basis for gain and loss of these compounds. And we have another story also why the other gene which is responsible or which is expressed in the wild strawberry, but what is not, it's not expressed in the cultivated one. So I'm not going to share that story, but I'm just wanting to give you the glimpse that how we understand how these molecules are synthesized. So coming back to India, we thought we will work on the mango, but it was not so easy because as far as the strawberry is concerned, and I'm sure you will agree with me that most of the strawberry has a typical, I will say, taste, and I do not know much of aroma that you realize. But if you think about mango, whether it's a taste or aroma, it's completely different. And when we started looking into the mango, it's not only taste or aroma, there are different size of the mango, different colors, and so forth and so forth. And this was very challenging. And we were like thinking, okay, it's a very important plant. Of course, it is originated in our part, Southeast Asia. It's the oldest cultivated tropical fruit plant. We have more than 1,000 varieties, and it is also gives important exchange. But if you think about this diversity, it's a bit challenging. So I'm not going to share all the data because this project we started almost 15 years back, and we have nice leads. Actually, we have 4 or 5 patents, cloning and characterization of specific genes which are responsible for making these volatile compounds, which we called it as a specialized qualities of hapus that we eat typically. So we perform, not only the metabolite analysis, not only NGS or recombinant protein, or we could also see what happens during the process of fruit development, what happens during the fruit ripening, and so on and so forth. I'm not going to tell you the whole story, but this is a classical method we used initially, GCMS based. And what we could see is there are very interesting compounds that, for example, monoterpenes are there, susciterpenes are there, lactones are there, and something called hexona and lonon, which is a very important component. And what is more important is also we found in some species even these pheromones, which are also have a very strong odor value. These are also synthesized. And this is the data I'm sharing from 27 mango cultivars, for example. So these are the different cultivars, and this is how you see whether it is a monoterpene rich or sesquiterpene rich. And now we'll consider only these first two, for example, monoterpene rich and sesquiterpene rich. You could see these varieties, whether it's Alfonso, Badami, or Chandrama, or Maya, Rajapuri. I mean, these are the different varieties. Neelam, I'm sure some of varieties you might be knowing it. So even I call them, or we call them, as a monoterpene rich, but still you see that in some varieties the sesquiterpene is also quite high amount, and even other metabolites are also very high amount. And that gives you a good understanding why each mango species has a different taste as well as different smell. And not only that, when we analyze some of the other varieties, for example, this most of these varieties except pyri, they are used as to make a pickle, and we could see that these varieties are rich in the sesquiterpenes hydrocarbons, and probably that gives better keeping qualities probably to the mangoes when we use it for pickle. So this is what, I mean, we started this analyzing. And one of the questions that we were asking, like, for example, hapus or alfonso. So although genetically we use these grafted plants, that means genetically they are same, but still we have a lot of variation within the alfonso, depending upon where it is grown. All of you know that we like hapus coming from Ratnagiri or Devgarh region, but same hapus, if you grow in Pune or even Usmanabad or even other places, you don't get such aroma. So that was one of the questions that we were asking. So for that, we did a, I will say, metabolite analysis, or NGS, like transcriptomic analysis, as well as proteomic analysis of different stages of developing fruits, as well as different stages of ripening fruit. And we could really, I will say, got a lot of interesting data that what happens during the fruit development, what happens during the fruit ripening, and we could build a lot of things. But I will share you a little bit excitements in this area. For example, so when we looked into the 3 regions, for example, these regions, like for example, we collected the fruit from Dapoli, we collected fruit from Ratnagiri, and we collected fruit from, I will say, Vengurla, which is close to Goa. And within this, like this is within the like 500-kilometer region. So, for example, Dapoli comes somewhere here, Devgarh comes somewhere here, and Vengurla comes somewhere here. So when we analyze this in more detail, we could see that the foods which we collected from the Apoli regions are more of a monoterpenic. That means the terpene content is relatively high. This is compared within the Apoli, Devgarh, and Bengal region. On the other hand, when we looked into the fruits which are collected from the Devgarh region, they are more rich in the lactose. And I think we got a particular answer because why we prefer the yogurt fruit over the Napoli fruit. And then recently we also did a detailed analysis of three selected varieties. For example, Alfonso, which is rich in the lactose. For example, the Pyri, which has a low level of lactose, and this foreign variety called Kent, which has no lactose. And it was very interesting and very exciting to see that how this programming of lactic biosynthesis happens in mango cultivars. And we are still under, I will say, investigation that how these compounds are actually synthesized. Although most of the genes are there in all these three varieties, but their expression is so much nicely programmed that Alfonso has ability to produce high lactose, whereas Pyri doesn't, and ketone even cannot produce this lactose. So this is a beautiful thing, and that's what I feel as a science or as a researcher gives you a lot of excitements, that you understand how this happens at molecular level, how this happens at gene level, how this happens at function levels, and so on. So now we are characterizing these individual genes. For example, we have patented this gene called enone reductase or methyl transferases, which actually use this mesaferon, which is having a very good properties. And how we are doing, as I showed you in this case of strawberry, that we are taking these genes, putting into the E. coli or putting into the yeast, and we are trying to see that okay, whether these genes are specific or these genes have a very broad specificity. And if they are like very specific and they are coming from hapus or mango, then we are trying to patent that these are the very important resources that has a great application in synthetic biology or chemical biology in Africa. So the other plant species that I'm going to share with you is Osimum, which is actually, I mean, everybody knows Osimum. I think it doesn't need introduction. It's coming from Laminaceae family. It has I mean, I will say very important aromatic compounds. It's a shrub. I mean, till this point, we know that it has a lot of kinds of secondary metabolites, particularly monoterpenes, particularly terpenoids and actually phenylterpenoids. Probably the site of synthesis is either leaves, because we do see them in the leaves. We also see them in the trichomes, which are the, I will say, hairy-like structures that are present on the plant parts, whether it is stem or whether it is a leaf or whether it is even flower. And we also see there are several compounds are produced in the even in the roots. And when we apply these various methodologies, we could understand that these compounds are very unique and very specific. For example, all of you know, for example, this is called camphor basil, Osimum kilimanjaricum. This is American Osimum americanum, or this is Osimum basilicum, or this is Osimum teniflarum, or earlier we used to call it as Osimum santum. There are a lot of applications are well documented. And what people probably did is taking the leaf extract or flower extract or root extract and then look, but nobody has looked into a specific compounds probably is responsible for this activity. And it is will be nice to also mention here, maybe it is not a very specific compound, although in several cases we have already evidence that the specific type of compounds might have a very high activity against specific thing. And I think I have given an example in my introductory slides, whether it is taxol or whether it is artemisinin or whether it is a morphine or nicotine or this aspirin and so on. So it has been already proven that these specific compounds has predominant activity, although there may be some synergistic effect. And maybe it's ideal to use a multiple or more than one compound for such activity. But I think unless we try to nail down that which compounds are actually giving a specific activity, or I will say kind of group of compounds, then it will be like becomes, I will say, our contribution to this area. So what we did is, like for example, in a very first phase, we try to understand whatever the species that we have. We have 10 plus species in with us that we are growing and we are analyzing. we could see that some of the species are rich in, again, these are osmium species I'm talking about. So some of the species are rich in the monoterpenes, some of the species are rich in phenylpropanoids, and some of the species are also having a combination of monoterpenoids as well as sesquiterpenes as well as phenylpropanoids. So just to tell you briefly, for example, this is again a review that we published 5 or 6 years back, that for example, these species like Osmium kilimanjaricum, Osmium basilicum, and so on, they are very rich in the, I will say, monoterpenes, whereas the green, for example, if you see Osmium teniplorum or Osmium gratissimum, they are rich in the phenylpropanoids. And there are some species which has even combination of monoterpenes and sesquiterpenes and even phenylpropanoids. So that is how that diversity do exist even in the within the species, and one has to understand this how we can further. We also looked into their trichomes, for example, whether it is a glandular trichomes or non-glandular trichomes coming from leaf or coming from stem or even coming from even flower parts and so on. And we could see again these are the very nice tissues that we can analyze because they look like very clean, because if you take a leaf, probably a lot of other metabolism also happens here, and your data could be sometime, I will say, not so clean if you want to really look into the metabolites. So these hairy like structures what we see on the different parts of the plant could be a very nice source to understand the metabolic diversity. Now one can ask me this question, why they are producing this organ or this particular hairy like structures. Probably these are the structures which are directly exposed to the environment, and the first line of defense starts from here. The way we put our soldiers on the border, probably this is the reason why these trichomes are present. They are very rich in the metabolites, which is probably their weapons to fight against biotic or abiotic stresses that they encounter when I mean, they have to grow and survive and sustain in the particular atmosphere or particular environmental conditions. So just I'm going to tell you the little bit story that me and my colleague Dr. Tulsiram and Dr. Mahesh Kulkarni, we develop a nice assay system based on mass spectrometry to understand, because in diabetics there are two major concerns. One is the level of glucose that goes up and down in the patients, for example, a diabetic patient. I mean, here I'm talking about mostly type 2 diabetes. And other complications happens in the diabetic patient is called HbA1c, which is glycation. So we thought there is a lot of studies available, and so we focused on glycation inhibition. So we wanted to look in this osmium species whatever we are using, is there any species has a molecule which can inhibit this glycation. And what we see, I mean, I'm not showing you all the data here, but for example, this is Osmium kilimanjaricum, Osmium teniflorum, and Osmium gratissimum. We could see that Osmium gratissimum has a high potential, whether these compounds are coming from leaf or inflorescence, that uh, that they have is actually a strong activity to inhibit this glycation process. When we looked into the more detail, like for example, what is the major compounds present in this Osmium gratissimum leaf or inflorescence, so we could synthesize or we could get these compounds, various compounds, and then we checked again the similar glycation inhibition assays. And what we could see, this is a standard this aminoglycan, but then what we could see, this a particular compound called eugenol is actually have a high potential to inhibit the process of glycation. Now you will ask me, what is the process of glycation? So glycation means this glucose which is present in a high amount can go and bind to the other protein. And the most of the complications, whether it is a retinopathy or nephropathy or even other complications, happens because of the level of glycation or gets increased. And that's why we also measure HbA1c, for example, in the diabetic patients. So this molecule looks like to us, based on our in vitro studies, shows very nice potential that it inhibits or it competes with glucose to bind to the other protein. So it doesn't allow protein this glucose to bind to the other protein. So this is what quite interesting story. But then when we looked into the glycation inhibition, we could see that okay, this is, I mean, very nice that you don't really require very high amount to inhibit. So it was quite interesting. So we did perform some bioinformatic analysis, and we could now actually show that actually this eugenol competes on the same spikes with a glucose, and this doesn't allow glucose to bind to the protein, and that's how this diabetic complications happens, for example. So when the next step towards this is like to feed this to or to inject this to the animals where we have STZ induced diabetic conditions, and then we have vehicle control, and when we eugenol. So what was quite interesting is that the level of glucose, for example, blood glucose, were dramatically reduced in the animals where we injected eugenol, and that was very very surprising because our hypothesis or our data which we have, it's indicated us that it has a property to inhibit the glycation. But now why the glucose levels are coming, towards normal, and this was very surprising. And then of course HbA1c was also normal and so on. So then when we looked into it, we could found very interestingly that this eugenol is not only inhibiting the glycation, but it is also inhibiting this alpha glucosidase. And one can imagine this alpha glucosidase is responsible for digestion of carbohydrates in our body, in our small intestine, and that's how the glucose is released. So if you have this molecule which is earlier identified as a glycation inhibitor, is also having this glucosidase inhibition activity, and that is quite interesting, by the way. It's very very interesting. So based on all these detailed studies, we propose that this eugenol has a dual role probably, and this dual role means it actually reduces, or it actually have a slow release glucose because it inhibits the alpha glucosidase, and at the same time it also actually inhibits the glycation, and that itself is actually a very interesting molecule coming from the osmium species having the dual role. And this is what I will say further histological experiments done on this molecule where we have eugenol injected, and other respective controls, whether it's a pancreas or kidney or liver and brain. We could see there's a nice recovery because of this eugenol, or the kind of abnormalities we have seen in the vehicle control or SGL induced uh, models. We could not see this when we injected eugenol. Also, we have a lot of in vitro data based on mass spectrometry, that is LCMS analysis or Western blot analysis, showing that yes, indeed, this eugenol is actually inhibiting the glycation process. And that was quite interesting. If you see very nicely, clearly here, that it does shows a specific, I'll say, glycation is inhibited by this. And based on that, we propose this dual role. That means this osmium metabolites, which is one of the promising molecule came out of it. It has a dual function, that it inhibits the alpha glucosidase, and that's how you actually slow down the glucose release. At the same time, this molecule competitively binds to the serum albumin, and it inhibits the advanced glycation end products, which are actually creating a lot of problems in the diabetic patients. We call diabetic complications. So now question was, this eugenol is not water soluble, and I think being the pharmacy background, the bioavailability of this molecule is a big question. So being a chemistry lab, we started interacting with the chemist, and we could now have more than 50 different derivatives from eugenol. These are the reported compounds that we synthesize in the lab. These are the different eugenol derivatives, and which has a different properties, for example, and we could produce in the gram scale. These are the, I will say, reported compounds, and these are novel compounds that we have synthesized and now we have patented. And the good part of some of these compounds, which have shown, like in the, for example, in the green, is actually showing a very nice the properties that we are expecting. So this is how one can take further, and now we are in process that, okay, whether we can develop some lead molecules further, which has a increased solubility and bioavailability issues, and can be used as a potential molecule which can take care of, say, diabetic complications. So that's what we are doing. Whether I don't know whether I will be able to finish this my presentation. So in between, I put this this acknowledgement. So of course, whatever the work that we are doing, although I have not shared you tomato or potato work yet in detail, as well as syropia work, but then I have shared mango and strawberry work and osmium work. So these are my collaborators, for example, coming from NCL. I mean, these are my PIs like Dr. Vidya Gupta, Dr. Tulsiram, Dr. Mahesh Kulkarni, Dr. Sachin Agawani, and Ravinder Kontam. These are the scientists, and all these are like students who were involved into. Most of them became a doctor. That means they got PhD from this work. We have a very nice team from Bahasaheb Kokon Krishi Vidyapit, Dapoli, Dr. B.P. Patil, Dr. Pujari, and Dr. Jamble, who help us to work on to got the tissues and to give a lot of scientific inputs for mango project. For potato and tomato, and even for strawberry, I worked with Asaf and Martin Jongsma when I was in the Netherlands. And right now we are working on potato and tomato. For osmium, again, these people are actually very much part of it. We have a team from Savitribai Phule Pune University, Avinash Kamble, then Abhijit Kulkarni, Tamane, Kotkar, Barokar, and these are the two students, Santan Panda and BK Shinde, who actually contributed greatly. And again, these are my students who got chance to work in my lab for their PhD on various systems, and they are now doctorate at various places, and these are currently with me. So this is my division that I thought I should show you. And this is one of the interesting things that I would like to show you is, so most of our students are our actually workhorses, and they contribute. So and this group, plant molecular biology group, was started by Professor Ranjekar in 1977, which was continued by Dr. Vidya Gupta later on, and currently we are 5 people. For example, myself, then Dr. Narendra Kadu, Dr. Rakesh Joshi, Dr. Kirti, and we have another scientist, Dr. Archana. So we are 5 people working at the moment in plant molecular biology group, and we work like a family. So it's probably somebody was telling me day before yesterday that I mean, from 1977 till today, we behave like a giant family as a group. So all these students are our students. It's not like my student or Dr. Narendra Kadu's students. So, and that you know that the benefit of joint family. So the students get a lot of benefit as well. We also get. So maybe I wanted organizer to tell me whether I should continue or should I stop to have a question answer session.
Have the slides are completed, sir?
No, it's not. There will be another maybe I will say two, three aspects that I thought I will talk to you, but I don't know. I want to have a question answer session quite nicely.
Okay, sir. Okay. Should we start for the question answer session?
Yeah. And then if you feel that we can continue, I mean, I have no problem to continue it further.
Okay, sir. So, looking in the chat box, our participants have also benefited by the knowledge they have got from you. While the session was going on, some participants were asking questions regarding today's topic. We will pick up few from them. So, first question is, how the biological activities of secondary metabolites can be studied?
Yeah. So I think very recent example that I gave is like, for example, if you remember the slide that I showed you on osmium, and I will go back to that slide. Is it okay?
Okay, sir.
Let me see. Could you see the slides or no?
Uh, yet not, sir.
It's not there, or it's
Uh,
there?
it's not there, sir.
Wait.
Yes, yes, yes.
Is it there?
Yes, sir, yes, yes.
The question is how to study the biological activity of
Yes, sir.
secondary metabolites.
Yes.
So if you think, this is what we have did. So for example, we have some idea that tulsi plant has been used, or it has some application in the diabetic management. So what we did is, here we took the extracted the specialized metabolites from the leaf and inflorescence of this Osimum species. In this case, for example, Osimum kilimanjaricum, Osimum teniflorum, and Osimum gratissimum. And we set up an assay. So to set up this assay, it took us a couple of months because this is a mass spectrometry based assay to see whether this compound has any glycation inhibition activity. So how you do that? It's very simple. For example, suppose you take a one protein which has a molecular weight about 5,000 Dalton, and if you add a glucose into it, okay, so if a molecule is getting this glycation, that means glucose is getting attached to it, its molecular weight will change. So it will be, instead of 5,000 Dalton, it will be 5,000 and additional mass of the glucose. Okay. Now, if you have a leaf extract or inflorescence extract or any molecule, you add into it, and if you want to understand whether this is inhibiting the glycation, you will be saying that okay, the mass increase that we see in the protein is not happening, and based on that, we will say that okay, how much, and this is called intensity bending MALDI, and that is what the assay that we developed in a couple of months, and then you can take even pure compound and do the same experiment. Of course, most important thing for that is you have to have positive and negative control for it. And this is one of our positive control, for example, a molecule which is known that it is responsible for glycation inhibition. And then these are our molecules. So this is how we test whether this is in vitro. Now further is actually I also showed you in vivo also. Like for example, the second property of this molecule which we came to know by accident is like it does also have glucosidase inhibition assay. So this glucosidase inhibition assay, because we work on amylase and amylase inhibitors and so on, or trypsin inhibitor, these assays, enzymatic assays are well established. So now you again add eugenol into it and you could see whether your enzyme is inhibited or not. So there are multiple ways to trace the I will say the biological activities of a particular molecule. So it depends upon what exactly that you are looking for. What is, I mean, what could be the potential targets? Sometimes it could be very simple, like what I showed you a minute before, like glucosidase inhibition assay. So you know that you in your lab you are doing routine glucosidase estimation. If something is inhibiting, you should be able to really monitor it. So that's how we do. I will say in vitro activity. Once we get a leads, then we can take candidate molecules even to do the animal feeding experiments. But now with ethical approvals and so on, it is very mandatory that we have very significant in vitro data. Then only ethical committee, this animal ethical committee will approve your project. So since we, out of hundreds of molecules, we are saying that we want to do this animal studies on a molecule which shows a very nice potential with our in vitro data. And then the ethical committee agreed, yes, it has something interesting. So let's perform. So based on that, we could now see that it is actually inhibiting or it is also in vivo inhibiting this alpha glucoside activity because we see less glucose. Not only that, we also see that the glycation is also inhibited. And then now the question was further like, now, yes, this molecule looks very nice, promising, but it has a problem of water solubility. That means its bioavailability is an issue. Now, how you do this? So how to increase the the solubility or bioavailability? So we modified this molecule in a various ways. So we developed more than 50 derivatives of this molecule, and then now again screen for all these in vitro activities. And among these molecules which has a better solubility and bioavailability properties based on our in vitro studies, now we are planning for animal experiments. And now we have patent for all these molecules which are promising molecules based on our in vitro and in vivo experiments. So potentially this becomes a drug molecule. So it's a process. So probably this experiment we started in 2014. And today we have, I will say 6 molecules which we called it as a lead molecule, which has a better means water solubility or bioavailability properties compared to the native molecules which are isolated from the plant. So it's a new chemical entities that we are coming, but based on some information which is coming from the plant metabolites. I hope I answered this question.
Yes, yes, sir. So next question is, can you please tell us something more about SGA profiling?
Sorry?
SGA profiling. You
Oh,
have
yes,
shared
SGA profiling.
insight.
Yeah,
Yes,
okay.
yes.
Very interesting question. So again, I will go back to the slides that I was trying to show you. Okay, this way I get a chance to do it further. Okay, so this is a question about HGA profiling.
Yes. Yes.
If you can imagine, these are the different on this. I don't know whether you will be able to see these molecules. For example, the first molecule I will read for you. It's called tomatidine 4-hexose. This is called hydroxytomatine, and all this. So for each molecule, we have analyzed on UPLC QTOF mass spectrometer. Okay, so you can imagine that to develop this process, we have to take help of a chemist, because I am not a very great chemist. I will say, being a biochemist, and even the lab where I was working, they were also good in the biochemistry and molecular biology, but not very good in the chemistry. So we developed the actually, I will say, a method for several of these metabolites to identify on LCMS. And again, LCMS or LCMS is not easy, because most of the mass spectrometry is based on the molecular weight. And there is a high chance that there are multiple molecules having exactly same molecular weight, whether you go 164.72, for example, this is the Dalton. So there is a possibility that there is another molecule which is having exactly same molecular weight. So that will not give a very good idea. So there is a nice tool in the mass spectrometry which is a bit advanced, which is called MSMS. That means you apply a collision energy on this molecule and you fragment that molecule. And then fragment of this molecule could be unique. And if you go back and remember, there was something called RFLP or DNA fingerprinting. So there we used to digest with restriction enzyme, for example. Here we apply the chemical energy to these molecules and we fragment. And this fragment could be unique for a specific molecule. So that means if you have a molecule or two molecules having 164 Dalton mass, and if you fragment, there is a rare possibility that they will generate similar fragment if the molecules chemically they are different. And these fragments one can use to identify, not only identify but also quantify. For example, in this slide, this is called heat map analysis, by the way. So this is the relative abundance of a specific metabolites. What you see in this, I will say, vertical names that I'm trying to highlight. So red means they are present in a very high amount relatively. That means like extreme red is, I will say, 100%. Something like between red and black is like 20%, and if it is a blue, it is not detectable. So now you take a very first molecule. This is tomatidine, for example. So if it is very high in the peel of tomato, which is actually coming from this small tomato, which is called immature green, whereas the levels are bit, I will say, high in the peel when this is a mature green or if it is turning brown. But if you see, the levels of these compounds are very, very high in the seeds. Okay, whether it is immature green or mature green or brownish or orange or red, the levels of these compounds like tomatidine are very, very high. So that way we can do this protocol. So that's why this work took 1.5 years. So in one slide. So this is a work of at least, I will say, 2 to 3 researchers for 1.5 years to establish this heat map. So research is not that simple, I will say, or we cannot really do something time bound. So this image, it looks like one image, but this was an effort of, I will say, 2 to 3 researchers, and including me. In fact, we did even further analysis what happens during the, I will say, different stages. I mean, this is a simple example. Like, for example, in this tomato, you have these kind of fruits. In this kind of tomato, you can have this kind of molecules, and this fruit has this molecule. So we could do even more detailed characterization. So this is I mean, this for this you need a good chemistry background to understand how one can develop a assay system or methodology, not only to monitor the parent molecule or native molecule, but its fragment. And then based on their intensity, because the fragmentation of a particular molecule could be unique. And then you can use 1 or 2 fragments or 2 or 3 fragments of each molecule for its identification as well as quantification. So it's a process. and it takes substantial time. But if it is a new molecule, to develop a method for LCM SMS based on I will say fragmentation, it takes some time to develop this.
Okay. So next question is, you have explained about strawberry flavor. So you have used GCMS. So what is the reason behind that using GCMS? There is a specific reason behind using GCMS?
A very nice question, by the way. So again, I mean, one can analyze the same molecules on even LCMS or other techniques. So in case of strawberry, we were looking for a specific compounds which are volatile. That means they are aromatic compounds. That means it has a actually property that they can be volatile molecules. And the interesting thing is, it's not all secondary metabolites can become volatile molecules. There are particular one of the criteria is that it has to have a low molecular weight. Some molecules which cannot naturally become volatile, we can derivatize them and make them volatile. So such molecules can be analyzed on GCMS. The reason why we use GCMS, because it's more easy, because you don't have to do the fragmentation. Because we have libraries, like we have 3 lakh compounds in the library. There are 2 libraries like Willy and I'll say this another library where we have 3 lakh compounds in the database. So if you analyze and you can identify without doing any further complicated mass spectrometry, you can identify whether the compounds which is already there in the library among these 3 lakh compounds, whether there is any match with your compounds that we see in the strawberry. So the reason is because we know the linalool and neraldol are small molecules. They have a property to become volatile, and for example, and then it's easy to.
Okay.
Yeah.
So next question is, plant specialized metabolites which are extracted that can be consumed directly or not, like artificial flavors which are available in market, they are
Yeah,
edible
yeah.
ones.
I already explained you one of the beautiful thing about nature or the enzymatic processes. They don't have this chiral mixture. They can make a very specific chiral forms. And I think again I showed you in the one of the slide, which maybe I can share one more time, is this one, for example. Yeah, you could see the slide.
Yes, sir. Yes.
Yeah. Okay, so I think you see in this slide, I think I showed you very clearly that if you see, if you try to synthesize, take an example of this linalool, which has a good aroma, for example. If you do chemical synthesis, there is a chance that even part of it, maybe 1% or 5% or sometimes 20%, it will have a combination of both, or LSS, whatever the chemistry that you do. But if you use the enzyme, which is actually this enzyme is actually coming from the gene which is actually isolated from the strawberry, and we have put into the E. coli and got a recombinant protein, and we provide a respective substrate. You could see exactly what happens in the strawberry is actually happening by this enzyme, and that is the beauty of, I will say, enzymatic processes. They cannot produce other kind of products. So now your question is, if you use the synthetic compounds, there is a chance that you might have 10%, 1% of this, and there is a possibility, for example, yes, linalool is safe because we eat strawberry and nothing happens, but there is a good chance that our linalool could be not good for us. And if that compound is there in our synthetic uh, 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 colorants, or I will say flavorants, it could be toxic or it could have adverse effect. Okay, so I
Okay.
hope I.
Yes, yes. So next question is, understanding of gene expression pattern will be able to identify the secondary metabolites?
Yeah, so uh, yes, of course it is possible. Let me also share some slides. It's possible. You see my slides, yeah?
Yes, sir.
Yeah. So for example, this is the what I showed you earlier, this heat map. So I now make it, I have made it a little bit smaller. So again, it is the same heat map where we were like looking for different metabolites in the tomato tissues. Now, if you see, if you do the real-time analysis of this gene, which is we call it as a game one, which is one of the glucose transferase, and another is a game four, which is a cytochrome P50. If you see broadly, I don't want to go through in detail, but if you see this red color also match with this metabolites. This red color also which match with metabolites. Red color is also match with metabolites. So that means these genes, game one and game four, are actually responsible for either of the steps in the metabolic pathway. So yes, it is possible to really, I will say, identify the gene which is responsible. It is not so easy. It is a bit complicated because so there are hundreds or thousands of genes which are expressed in the specific tissue. I mean, if you think about this slide, I said that bioinformatic tools are very important. For example, now there is something called co-expression analysis or network analysis. So if I know that game one is actually responsible for making a specific steroidal glycolide, I can use this game one as a bait molecule and use the transcriptomic data set available for all this tissue, and I could see that okay, how many genes are actually co-expressed with my game one. This is also possible to do with another gene called game for example. So I need now there are 89 genes which are expressed. So that way you can actually shortlist from thousands of genes, you can come up with 89. So now in this slide, what I'm showing here right now is actually even much more complicated because we use game one and game four from tomato and game one and game four from potato, and we ask the program to identify which are the genes expressed co-expressed, I mean very high chances of co-expressing together with this. So we got this big number 257, 13. This was another, I think, 1000 or so. This is 89. So it was very difficult to characterize all of these genes. But when we did much more complicated bioinformatics, like looking for game one, game four from potato and tomato respectively, and asked the program to find out which are actually common for all four of them, and then we came up with a dozen of genes. And this is what one can actually understand how the genes, how the particular metabolite is synthesized in a particular tissue, and then we can understand the biosynthetic pathway, which actually, and then we can take some of these genes and characterize. And how to characterize is also a bit challenging. For example, I will show you one slide. So, for example, if you want to understand, and now you know that game 7 could be involved into the this biosynthetic pathway, but how you will prove it? It is always challenging to do the enzyme assays what I showed you for strawberry, for all the things. So here we ask this question to the plant, and how you ask this question to the plant. For example, we know that in tomato, I know that tomatin is produced. So now I can silence this game 7 in the tomato plant, or I can overexpress the game 7 in tomato plant. Okay, that's nice. You established a plant where we have game 7 overexpress or game 7 silence. So what will happen if I silence the game 7? So this reaction, enzymatic reaction, will not happen. That means conversion of cholesterol to 22 hydroxy cholesterol will not happen. Means what will happen? We will be not able to see this compound. And when you do mass spectrometry, we'll see that this compound is reduced, because based on how much gene is silenced, it is completely reduced or it will be partially reduced. And what also could happen is that this compound will be accumulated. So that is how you can establish the function of the gene. I will give you another example. For example, if I want to understand what is the function of game 8, again I can do the silencing and ask the question to the plant instead of doing this recombinant enzyme. We can silence this gene or overexpress this gene. And if I overexpress this game 8, what will happen? this will be completely gone, and this compound will be higher, and probably this whole pathway will be higher. Like, I mean, maybe I will see increase in alpha tomatin level. We may not able to sometimes detect these intermediates, but at the end we will see that these tomatin levels are increased. If I silence this game 8 again, this tomatidine will be less, and this compound, probably 22 hydroxycholesterol, will be very high. Of course, I want to tell you that life is not so simple. Sometimes something else can happen. If you see here, for example, if I silence game 4, we know that this whole pathway got shut off. But unfortunately, we did not see this compound is accumulated. And later on, we could realize that this compound is used for another pathway, and that is what we call metabolic flux. That means the flux which was coming for this pathway was being changed to this pathway, and we could see steroidal saponins are produced. So that is, I will say, uh, also could be a complications, and one has to understand how we can correlate whether this particular gene is expressive or responsible for this metabolite production or not. So it's sometimes it could be simple answer, sometimes it could be much more complex answer, and sometimes you might not get answer right. That's also possible. That's a research.
So next question is, plant stress hormones and its signaling mechanism in plant can be propagated in humans for the management of stress-induced conditions.
Yeah, the probably the hormones that are present in the plant and the hormones which are present in the, I mean, I will say not all, I will say, but several hormones which are very specific for the plant, and there are several hormones which are specific for, I mean, human beings or even other organisms. So to answer this question, partly yes, partly no, because if you take something and which is actually precursor for some hormone, so it becomes a substrate available for the hormone. So that's why a lot of things, I mean, whatever we ingest, for example, whatever we eat, we don't use it directly. We metabolize in a way that we want, or the way enzyme system is available. For example, you know, when we eat a carbohydrate or maybe starch, a lot of things happens in our saliva. There is a plenty of enzymes, like for example, salivary amylase is known, that it digests and you try to taste it as a good. By the way, starch is not I will say sweet, but if you digest the starch and convert it to the monosaccharides, it tastes as a sweet. So this happens probably in our mouth, and then we say it is a sweet. If you directly eat a glucose, you might not have to do that. So same thing happens, that several of these plant metabolites which enters in our body, it's not directly used. Some of them serve as a, I will say, basic components like nitrogen, or I mean, whatever the carbon or phosphorus, and some of the metabolites are used to synthesize some molecules, and it could be hormones as well. So yes or no, that's what I said. It depends upon what. For example, vitamin C we take as ascorbic acid. For example, it comes from the plant source, and yes, it does serve the purpose of vitamin C, but not for probably the hormone which are very specific for our humans or plants.
So last question is there any database available that indicates whether a given enzyme contributes to the primary or secondary metabolites?
Yes, there are several databases, and one of the, I mean, so cake pathways, you know, there are like some programs which can tell you that okay, this pathway belongs to a particular, I mean, this enzyme belongs to a particular pathway or not. So the one of the major challenge there comes is the isoform complexity, and that isoform complexity makes life much more difficult. And again, for that, I will show you one slide, and I hope can go quickly to the presentation. Could you see the slide?
Yes, sir. Yes.
Yeah. So, for example, this is a one of our paper published in Nature Plants in 2017, I think. This is the metabolic pathway starting from, I will say, melanate to cholesterol. That how it happens. So, if you see, these are the some of the enzymes. By using the databases, we could see that they are actually involved in this metabolic pathway. So say example, for example, this enzyme C14 reductase. So based on their sequence, we could see that okay, it is a C4 reductase, and even further sequence analysis could actually demonstrate that it can use this locurol as a substrate and convert it into this molecule based on the, I will say, bioinformatics tools. But if you take another example, take this example of SMO2, this is oxidase. And based on the sequence analysis, we could understand that it has oxidase activity, but you could not understand whether it can take this as a substrate because there is another SMO which is also there. So for that, to understand the function, then you have to do some enzymatic assays, whether it is cell-based assay or whether it is a recombinant protein assays, or if you can ask this question to the plant by silencing and overexpression, then you can say, yes, this SMO4 is actually very specific for this substrate and produces this, but it's not a same enzyme that can take that can function as a SMO2. So, or we take other example, for example, this desaturase. So definitely the C5 desaturase here is different than the C5 desaturase homolog here. So you get some answer, but to understand in a very detailed way, one has to really think about, understanding their function in much more complex way, and it takes again substantial time and substantial ways to demonstrate this. It's not a very simple answer, yes and no.
So, thank you, sir. Thank you very much. And thank you all the participants. There are n number of queries and n number of questions of the participant, but due to time constraints, we will stop the question answer session here. But I hope all the queries have been resolved by sir, for sure. They have explained it very beautifully, the plant metabolites concept, and we will understand, we have understand it very well. So thank you very much.
Thank
I
you.
would
Thank
like
you,
to give,
Shobha
there
madam,
were,
for giving me this
I would like to,
opportunity to talk.
yes,
Yeah.
I would like to give a huge thanks to Dr. Ashok Giri sir. And now I would like to request Dr. Shobha Tole ma'am to present a vote of thanks. Please, ma'am.
Yes, I'm audible, sir.
Yeah.
Yes.
Good.
Okay.
Yes,
Okay. Chitra, I'm audible.
yes, ma'am. Yes, yes.
Okay.
Please
So,
continue.
myself is Dr. Shobha Tule, Associate Professor in Medical College of Pharmacy. So, firstly, I invite Dr. Ashok Giri sir, okay, to spending a lot of the times on the behalf of other Shikshan Prasarak Mandal, Usmanabad, on the occasion of the beautiful scenario of our first EFDP program in the academic year of 2022 in a platform of online mode in a Category College of Pharmacy on the which is entitled as the Herbal Medicine Research and Global Health, a therapeutic perspective. So, sir, as you have to dealing plant specialized metabolics in healthcare. So, regarding this, I'm firstly saying that you are the dynamic personality in the world of the biochemical sciences, and it is through by your presentation. Your presentation is very informative. Your slides are very informative, sir. That can be showing, and also my upgrading the knowledge regarding the as up to these sites. I only knowing that the primary and secondary metabolites regarding what are the tannins, alkaloids, okay, and what becomes the different roles. But no, also any idea the regarding the our holy plant osimum. It's a different varieties. It's a different aromatics which constituents are responsible for that aromatics, and also only up to this age, sir, I'm only eating the mango, but in a different varieties of the mango, why the taste and aroma is different, which constituents are responsible that, and as in also in a you are the such a good researcher. So what are the current aspect that you have to implement it in a day to day that I have observed that in a starting that you have to show in the one slide is that is in the current particularly got the research that is the paper is published in chemical sciences, okay, what becomes the importance of machine learning and artificial intelligence in crude drug analysis, and whatever the professions regarding also in the pharmacy the peoples are implemented in that to make the formulations, okay, and that can be applied, okay. So regarding this aspect the session is very beautiful, the session is very important, and session is also very knowledgeful. I hope that the every pharma people, the getting the good knowledge regarding from this scenario. So once again, I'm very thankful to you, sir, for to getting such a nice information, spending your valuable time, and within only a single telephonic call, you have to accept our invitations to join in our sessions. I hope also in future the NCL team and with you also the cooperated to our students belong to our rural area. Okay, to
Yeah,
make
very much.
also informative. Okay,
Thank you.
so okay, I'm also thankful to our honorable chairman sir, Sri Sudhir Patil sir, to upgrading the educational area in Usmanabad also. And also I'm thankful to our administrative officer, Sri Aditya Patil sir, day by day providing a good platform to our college faculty to make enthusiasm in research and also carry the various academic activity in this platform. Also, I am very thankful to our beloved principal sir, Dr. Amol Joshi sir, to make the faculty motivation, encouragement, giving the enthusiasm to doing also the research activity and make the implementation. Not only in this regular period, also in a pandemic situation. The sir is motivated to all faculty to doing on online mode, to doing the various jobs that are needed in academic sections also. Now, last not but least, I'm very thankful to the coordinator and also the vice principal of this college, Ms. Geeta Sapkar Madam, to motivations to every faculty members for performances of this scenario also. Now, I'm also thankful to cheerful organizing committee because of this committee, this whole event can be possible. Firstly, I am thankful to Umbre sir, Alkunte sir, Mali sir, and Chitra madam. No any technical arising can be happening. That is, as the sessions are starting from that is 9th of the February to up to this. Okay, it is ending up to 14th February. So the whole team is working very nicely to make the motivations to encourage us, all the researchers and doing the well platform for our college. So I'm very thankful to all. Lastly, I'm very thankful to all the delegates to listen patiently. And sir, again, I'm very thankful to you to giving your valuable time today. Okay, sir. Okay, over to you, Chitra.
Yeah.
Thank you, ma'am. Thank you so much for presenting vote of thanks, and thank you very much, Dr. Ashok Giri sir, for
Yeah.
sharing your valuable time and knowledge with all of us. I must say the session was very beautifully explained by you, sir, and we will be always grateful to you for your kind cooperation for the success of today's
Yeah. So we should
session.
not end. I mean, after thanks, I think we should not say anything. But I would like to say two things. One is, I think we should thank to all audience because they were very, I will say, very interactive. I mean, their questions were very, very relevant. And without them, I think we cannot organize conferences because no,
Yes.
whole idea is having active participation of the participants. So I think that's most important. I think in between there was some network issue, so probably Shobha Madam must have been thanked to participant. But the second thing I want to tell you, which is also very important for all students coming from our region, is it is not like you know how to study in a very good schools and so on.
Yes, sir.
So
Yes.
I studied in Jilla Parishad school till my 10th, and also do not have much background, but still I could I'm proud to say that we could do good science in institute like one of the best institute in the country called National Chemical Laboratory, or wherever we got, I mean there may be more than 100 students got benefited to go to the different countries and establish themselves in the field. So it doesn't, and I come from a even much more a poor district than Osmanabad, which is Bid. So it's very well possible. So I want to tell our audience that sky is the limit. It depends upon. It's very easy to sympathize that okay, I come from rural areas, but I think if you put a good effort, uh, I mean, focus on your thing, talk to the people, get expert, I mean, opinions, and get guidance from the people like what in colleges. I think you can have a great career, and I'm sure all of you will be very successful in your respective interests and respective areas. So I just want to tell you that it is nothing to do with your where you are studied. It is
Yes,
all
sir.
you can decide yourself what you want to do. So
Yes.
that's what
Your words, yes, yes,
and thank
yes.
you for all of you
Your
for
words
having
are
this.
always motivated to all of us, sir.
Yeah.
Okay, sir.
Yeah.
Okay, sir. And
Thank you,
second
sir.
time
Thank you.
listening that very important.
Yeah.
Thank Thank you, sir. Thank you once again. And I'm presenting this virtual certificate. This is our
Okay.
small effort. Yes,
Oh, I will
yes,
take it virtually.
yes, yes, yes.
Okay.
Thank you
Thank
so
you,
much, sir.
Miss
Thank you.
Chitra, for nice coordination.
Yes,
Actually, although you have some internet issues, but you could manage
yes, sir.
very well.
Yes.
Yes. Thank you, sir. Thank you, sir.
Best luck
Thank
to
you.
all of you. Thank
Yes.
you.
Yes, sir.
Thank you,
Yes.
sir. Thank
Yes. Thank you, sir.
thank
Thank you.
you. Yes. Thank you, ma'am, and thank you, sir. Thank you once again, dear participants. Now we will conclude our session and we will meet tomorrow on fifth day of International Faculty Development Program with new resource person having new perspectives. Tomorrow we will have Dr. Uday Devkate sir from Government College of Pharmacy, Aurangabad, who will orient us about the role of HPTLC in identification and purity checking of herbal drugs. So stay tuned and have a good day. Thank you.